Hi everybody, this is Mr. Nolan, and what I'd like to do with you is to go through what we discussed today and what we saw in class. There's some reminders up here on the screen that I'd like you to take note of in case you were not here. There's some things you'll need to be working on over the weekend. But the main point of this video is to show you how to do this computer lab simulation that we did in class, so that if you weren't here, you have this explanation to show you how to do this. What we're trying to do, if you recall, we have been learning about Duchenne muscular dystrophy, DMD, and we have been kind of asking the question, for one thing, can we do anything about it? Can we cure it? And we have also observed that it affects pretty much only males. It's almost unheard of to find females who have DMD. So these are the two things we're interested in. And so what we've been doing is we've been looking at pedigrees, Punnett squares, we've been looking at different kinds of inheritance, which you have a quiz on Monday. And we understand how these things are inherited. We see some patterns. What we have not talked about is what I usually refer to as the like the nitty-gritty chemical details. What are like the molecules that are all determining your traits? And so some of them you've heard of. DNA is one of them. This is involved. Your proteins are also involved. People are made out of proteins. And so DNA codes for proteins. You're made of proteins. So DNA, that's how, DNA is affecting our traits. Well, I want you to learn about this model that scientists have established or developed about how DNA ultimately determines your traits, whether you have dark hair or light hair, or your skin tone, or how tall you are, or eye color, any variety of other different traits, even certain personality traits might be affected by DNA. Your proteins in your body are really what are going to determine your traits. So we're going to do a little simulation online, and I want to show you this simulation so you understand how to do this. There is a reading that's associated with this that I have sent out on Remind, so you have that reading. But I want to show you how to do this simulator. So in order to understand how this model works, how does DNA turn into traits, we need the student activity sheet, and I'm going to show you what that looks like. It looks like this. It says Lesson 6. Why would a body fail to make a protein it needs? Because a person with DMD, their body fails to make dystrophin. And so what we're interested in is why. Why does their body fail to make dystrophin? So we can see here, if you look at my screen, we can see that there's a sheet here that is asking us to go to a certain website, and we're going to follow along with these steps. And I'm just going to kind of talk to you about how to do this. I'm not going to do this step by step. I'm just going to show you how to do it. So the first thing we're going to do is we're going to go to this website, and then we're going to answer these questions. Okay? So let's go to that website. If we go to that website, it looks like this. It opens up just like this. And to use this website, pay really close attention. The website is going to simulate DNA turning into traits in your body, and it also lets you do mutations. If you look down here at the bottom left, this enables you to mutate that DNA to see this is how this affects this gene. So this is a pretty cool model. It can be a little clunky, which is why I want to send this out and talk to you about it. First of all, we can choose what genes we are working with. It starts with lactase. This is the enzyme that lets you digest milk and dairy products. If you don't have functioning lactase, that means you're lactose intolerant. So for any lactose intolerant people out there, your lactase, it don't work. But what we're going to do is we're going to click on DMD, and this gene, these letters that you see right here, is just a part of a gene of DNA for the dystrophin gene, which if it doesn't work, it gives you DMD. So really, strictly speaking, this is a part of the gene when it works, right? When you don't have DMD, when you're healthy. So in order to make this simulator work and to see how this DNA turns into traits and what mutations do to them, we're going to go click on Setup here, right there. And we also need to click this little button that says Go Stop. If this is not highlighted, this won't work. All right. Okay, something else I'm going to do, just to make life a little easier, I'm going to click this little box that says Show Genes so that we don't see any of these little boxes. It just gets a little confusing. So what we have here is a strip of DNA. So if you look really carefully, some of the questions in your student activity sheet are going to ask you to make an observation. So make some observations right now about this DNA, and you'll have to do this on your own. So we see there's some letters here. We see there's certain letters. They kind of come in different colors. If you look really closely, you'll see each of the letters has a different shape too. It's pretty interesting. So this DNA is in your cells, and we're going to create a trait out of it. So all you have to do is just click these in order and follow along with the student activity sheet, which tells you what happens when you click transcribe, what happens when you click release. So we'll go through these right now together. You can even work on the worksheet as I go. You can follow along with me. So if we click transcribe, we see that there's new letters. There's new letters that are now attached to our DNA. Well, that's interesting. Again, if you look really carefully, you'll notice that the letters are a little bit different. Instead of T's, what do you have? We've got U's. We still have A, C's, and G's, but we've got these little U's instead of T's. That's pretty interesting. Well, this new chemical that is stuck onto our DNA, according to this simulator, is called RNA. It's similar to DNA, but it plays a few different roles. So let's click release. So we see this new letters has been added of RNA. Let's click release. Now the RNA has moved up. It's actually moving out of the nucleus and into the cell. So that's what happens when we click release, is that the RNA moves somewhere else. Let's click translate. This one's really exciting. Whoa! So we see a bunch of things have just been added. So these yellow things on the bottom have been added, and then all these labeled things on top have been added. If we click release, the yellow things disappear. I am going to talk to you about these yellow things for just a second. These are called tRNA. And tRNA stands for transfer RNA, and they've actually dragged all these things on top over, because when we click release, they go away. So the tRNA, that yellow stuff that was just there, that squiggly stuff, it's now gone, right? It doesn't have any role anymore. I want to highlight something and then talk to you about it a little bit. So what you'll see is that I've labeled the protein, I've circled the protein that our DNA has created. Our DNA made some RNA, and through the help of that yellow squiggly guy, that tRNA, we create some protein here. So you'll notice if you look carefully, again, you always got to look carefully. I keep saying that, but you really do. If you look at this protein, you'll see it's made out of these individual little units. It might be a little bit hard to read in this video, but you'll see over here we've got MET, LEU, TRP, TRP, GLU, and you'll also notice that those different labeled components are shaped and colored differently. Well, let me explain to you what this is. Our DNA has just created a protein. I said at the beginning of this video that your proteins are really what determine your traits. So this protein that we just created in our cells are doing this all the time, 24/7. This protein will give you dystrophin. Dystrophin is good. You need it for your muscles to function properly. And this person would have a healthy dystrophin protein. Their protein works. It's functioning. Now, in order for a protein to work, typically, it has to have a very, very precise sequence of these units. And if even one of them is just a little off, it doesn't work. We can see that example in something like sickle cell anemia, where sickle cell anemia results from one letter changes, and then our protein doesn't work. It codes for crappy hemoglobin. But anyway, that's a bit of an aside. Over here, what we can see is that all these different little units have come together to make this long strand of protein, which I have circled here. And each one of these is called an amino acid. That's what each one of those little three-letter units is called. It's an amino acid. Okay, so here's why this is all important. If you have DMD or any other kind of genetic disease, what's probably going on is that your DNA, you have received a mutation, a bad mutation. That mutation, if it results in a trait, we call that an allele, so that it's a different version of the gene. It's a different allele. It might be dominant or recessive. There's no way just by looking at the DNA to know if it's dominant or recessive or X-linked. But the important thing is to recognize that changes in the DNA, these letters, can result in a different gene, a gene that maybe doesn't work. So let's go ahead and try that down here. So if we look here at this mutations portion, we see we can select a mutation type. And let's say that we pick deletion. This is actually a really bad mutation to have. You don't want deletions. So we can also pick the number. Let's actually pick a good number. Let's pick like 5. So like, you know, your cells are doing stuff, they're creating this DNA, and all of a sudden you get 5 deletion mutations. This sounds pretty bad. So here's how to use the simulator now. click replicate the original DNA. And when you do that, what has happened is that we've created a new strand of DNA, but look, it's missing some bases. It's actually missing 5 of them. That's because we picked 5 over here. We also picked a deletion, which means it deleted them. They're missing. So this strand of DNA is what we have left over from the original one. Now, we could have picked an insertion, or we could have picked a substitution. The kind of mutation doesn't really matter. The important thing is that the DNA has changed. So, and then also if you look down here at the DNA sequence, you can also see that it's missing those letters. So let's go ahead and try to transcribe this and then see and translate it and see what happens with our protein, with our final product. So let's go ahead and transcribe. Wow, it won't even transcribe the whole thing. So it looks like we've got an issue here where it won't even transcribe the whole thing. Click release, translate, and then release again. So now, oh my gosh, this looks terrible. So if you look down here at this new protein, it sure looks different from the original one. So look here. If we look at this protein, we can see that the first few amino acids, MET, LEU, TRP, TRP, GLU, GLU, VAL, GLU, well, that one's messed up. Some of them are okay, but all of a sudden where our deletions come in, we see that the rest of the protein is gone. It's not even there, which means that this is a defective protein. If even one of those amino acids is different or missing, or maybe there's a new one, you stick in a new one, this protein will not work. If you're super duper lucky, maybe it will, but we're going to assume for the sake of this lab that if even one of them is different, the protein does not work. It's very specific how proteins have to be built because they have to fold into a certain shape. And so if they're not built right, they won't fold into the right shape and they won't work. So this person who has the defective gene, they will now get DMD. They're going to get Duchenne muscular dystrophy because that gene is broke. They're not going to make dystrophin like they need in order to have their muscles function properly. So we can do other mutations. We don't have to just do a deletion. We can do other mutations. So I want to talk to you about that really quickly, and then I'm just going to let you do this on your own. Go to this website. There it is. And go ahead and do your own work on the student activity sheet, and just do your best. You want to figure out these mutations. But let me just show you 1 or 2 more things about this. Suppose that I want to try a new mutation, because in your student activity sheet, it's going to ask you to do multiple mutations, like here, right? If you see on the screen, it says you want to do, for this part 2 for DMD, you want to tell the type of mutation, such as a deletion, so that's like what we did. And then the number of mutations we chose was 5. And the number of changed amino acids in the protein, we would have to count them. If there's any amino acids that are missing or different, you would want to count them. So if we go ahead and count these here, we'll see that there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 that are missing, and then 1 that's different. So that makes 11 total that are different. So for this particular example, there's 11 changed amino acids in that protein, and it won't work, right? It will not be functional because that protein is now different. But we want to do this several times. If I wanted to do another mutation, in order for this to work, what I recommend that you do is go back up here to Setup and click it. It will refresh everything. It'll give you your original DNA. Go ahead through, click transcribe, release, translate, and release. It'll give you exactly what you started with, okay, as long as you stick to the same gene. And now down here, this is where you can, maybe let's pick, a substitution, and we can choose a certain number. Maybe we want 3 substitutions or something. Now click replicate the original DNA. There's your new DNA. And then just go back through, transcribe, release, translate, release. And again, we're going to look here and we're going to see if we can't find any changes. So if we look really carefully here, it looks to me like this one, VAL, is different. It's now ASN. So we see that this is the one that has changed. I don't think I see any others that have changed. It looks like that's the one that has changed. So since that one has changed, what we would do is we would run over to our student activity sheet and we would say, okay, we tried a substitution. And we did 3 mutations, I believe. The number of changed amino acids was 1. And again, no, this protein will not work. So you're going to go through and do this whole table with, different mutations and different numbers. And as long as there's a change, that means that no, that protein will not work. So I hope that this was halfway useful for helping to kind of explain how the simulator works. We will have time, one more class session again for you to work through this if you need some help. But I do want you to get started this weekend if you weren't here. That way you can kind of start to play with this a little bit. I have sent you the student activity sheet. That way you can't tell me you don't have it. Okay? So have an awesome weekend. Good luck with this simulator. And I will see you next week.